So hi, hello and welcome again everyone, and welcome again to another Saturday Microscopy live stream. Microbehunter here. And I see that many people have already started to post comments in the chat, and would like to say hello all around the world. Of course, I would also like to ask you, like every time, please to give me a short feedback on the sound quality. I have to readjust the sound every time when I start the live stream, and as you can see, I'm wearing now a headset. So there's also a cable here. I'm going to put it on the side here, and yeah, so I hope that the sound works, and I would like to ask you please to quickly give me feedback if everything is fine, if it's too loud or maybe it's a little bit distorted. Yeah, I think the sound is good. I'm getting the first messages already. And you can also see that the setup today is a little bit different. I now included the desktop on the side here. The microscope view is here, and then there's another microscope view of what you can actually see through the microscope. So everything's a little bit more cleaned up now, and of course can also change the setting around so that for example, if you want to look through the microscope directly, that's basically the thing that you can see right now. So it's a live view as well. Okay, what I will do today is the following. In order to structure today's live stream a little bit, I'm going to break it apart into different sections. I'm going to show you today a little do-it-yourself project over here. This here is a very, very cheap and a very fun microscope to use. It was sent to me from someone who's working on this project. I put the links into the description. It's 3D printed, and there is a camera module here. So I'm going to be talking about this here as well. And you can see that I already put a slide in here. Very cheap and very fun project to use. That's number one. Number two, the second thing I'd like to talk about today is I would like to show you the app and the web page called iNaturalist, which might be also interesting for those of you who would like to identify the microorganisms that you see under the microscope. I think it's a very nice app. I don't know if some of you already know it or not, but I'm going to show that to you as well. And then, of course, like every time, I'm going to do a Q&A, a question and answer session. And I see that essentially that some people already started to post. So what I'm going to do right now is I'm going to just start talking a little bit about this. Then later on, maybe after about 20 minutes or 30 minutes, I'm going to go through the questions. So please post comments and post questions, and also tell me from where you are. Even if the questions are not directly on topic, that's also fine. Okay, I will try to answer them as well. And then I'm going to talk about iNaturalist, and then I'm going to do another Q&A session. It should kind of keep us busy for today. Yes, well, first of all, what I've got here is, as a matter of fact, 3 fun little microscopes. Here, 2 of them are already assembled. These 2 here, and this one is one taken apart. And here, this is just a power supply. So this is not a USB connector. Well, it is a USB connector, but actually it's not, the image is transferred over Wi-Fi to the computer. And what I'm going to do is I just put the slide on here, and I'm going to show you what you're able to see. So this is now the live, the view. Using this tiny microscope. You see it's a little bit blurry. I have to focus it a little bit, so you can use a little tool here to focus it by turning it here, and basically you can use this microscope to directly live stream things like I'm doing right now. But it's actually a camera module that's also used for home surveillance, and it's a standard camera module. And how much does it cost? It costs around €6 or $6. It's really cheap, and it's actually a complete it contains a Wi-Fi module and everything, and it's quite nice to be used here. So I'm going to show this microscope a little bit to you, how it's actually set up. And if you want to support the project, please do go into the description, and I posted a couple of links there. And if you are into 3D printing, then you can, of course, also 3D print those yourself. I got them sent, but they are also 3D printed. Look kind of nice. So what I'm going to do now is I'm going to show you a little bit how this project microscope is designed, and the question is of why would anyone use something like this if we have already basically full microscopes, right? And what's the advantage of this here? I think the advantage here, or the fun thing here, is it's beside the low cost, it's a fun project. So especially I am a teacher, and if I want to do some do-it-yourself projects with students, and this is certainly something that can be done. And the good thing is this camera here can be programmed as well. However, it does work pretty much out of the box, so there is no programming necessary, but you can program it if you want to as well. So what I'm going to do is I'm just going to show you here how this microscope actually is designed, and how it. So let me push this here to the side a little bit, because the focusing mechanism is quite fun as well. And what you have in here is of course the camera module, and this stage, the top part here. I'm going to turn this around here. If you press this, you can see that it's kind of, it almost is like a spring, right? And by tightening the screws, you're able to change the distance. But there is no spring here. There are actually magnets here, pretty strong neodymium magnets which push the stage up. And then using the screws, you can actually adjust the focus. Then my whole microscope can be assembled simply with this tool, so there is no gluing necessary or anything like that. Simply put everything together, and there are a couple of screws, and that's pretty much it. And over here you have a USB connector, and this USB connector is used as a power supply, of course. You can also, if you want to go a little bit more advanced, you can also program the camera this way. So for example, if you want to do time lapse, or if you want to program it to automatically take a picture every few seconds, then that's also possible. Or some people have used the camera also for animal watching, to automatically trigger automatically the shutter release. So all of these things are possible, and there's even over here on the other side, I can actually show you here on this module. Let's take this out here. Maybe it's easier. So here, this is the camera here. It's actually 1600 times 1200 pixels. It's pretty decent. 1600 times 1000 by 1200. It's a pretty decent camera. And there is an SD card slot here, which I don't need, and there is Bluetooth and wireless transmission. So the picture is transmitted wireless. So for example, what you see over here, go back here, this picture that you see here, that is actually transmitted wireless to my computer, and then I included it into the streaming software. How is this possible that this camera is actually able to magnify so much? And the reason is because in this microscope here, and I have to show it to you here up from the top part, you see that there is over here, there's a little bit of an extension here. Okay, so the sensor is down here, and because of this extension, the lens is raised away from the sensor, and this increases the magnification. So what you have to do is I'm not going to do that now, but what you have to do is you have to unwind, and you have to screw out the lens, and you have to connect the lens here, on top here, on top here, and then the sensor is on the other side. So there is, I don't know, maybe a centimeter or so. It's raised away from the sensor, and this increases the magnification. So it's actually quite a nice thing. And what projects do I already have in mind? Well, obviously, why not try to 3D print or make something where you can actually use this camera also as a camera for the regular microscope. So, for example, if you construct yourself a little case, I've been also 3D printing a little bit and experimenting with that, and then you can attach it to the eyepiece, and then you have a wireless transmission. And I've tried this, and the image quality is significantly better. Okay, significantly better even, because we're using actually the optics of the microscope to magnify, and then you're just using this lens over here, just to create the image. So the image, by actually pointing it to a microscope, is quite good actually. It's almost like when connecting a mobile phone. A mobile phone camera, it's also quite sharp. So basically, this is a very nice nice project. How much does it cost? You can buy this online over AliExpress, which is a Chinese supplier. And the camera module over here, just the top one, because you can detach it, costs €6. That's around $6 or $7 US. Just this one over here. And this one is the main board, so to say, which contains the USB connector. and this one costs, I think, also around that much. So it's fairly cheap. However, you can, I think if I'm correctly informed, if you connect the voltage directly to the correct pins here, you might not even need this part here. But this part is needed if you want to actually program it, but there is no programming necessary. Yeah. So this is basically how this, we had a little, it's called the Matchboxscope because it has the size of a matchbox. It's an open source project. And it's used, yeah, and these are readily available parts, so there's nothing custom. It's called the ESP32 cam, which is a fairly popular module that a lot of people are using to do all sorts of projects. Time lapse, whatever. Yeah, you can essentially set it up somewhere, and then you can program it to take a picture every couple of seconds or every couple of minutes, and basically forget about the camera for a week or so, and then you have all of the pictures. Yeah? Or if you want to do some surveillance, and if you want to yeah, transmit the image wireless. Yeah. So this is essentially something that yeah, is kind of fun to experiment. And what I would like to do now is I would like to just show you a few slides using this microscope here. Okay. Oh no! Before I do that, I just want to show you a little bit how the light actually works. This is because the light is quite fun. Yeah, what it's got here. This here is yellow. This yellow thing that you see over here. Yeah, that's an LED. All right, and it's pointing upwards. Yeah, and what happens is the following when it's in here. Up. I have to take this out here. There is a little hole in here, right? And the light is passed through this hole, and then you see this little contraption over here, which is painted silver. So the light goes out and simply bounces around on the inside and then is projected down again. So that's a very simple but effective way of solving the problem. So even though the light comes from the bottom, it's kind of redirected yeah, down here. Maybe you see it over here a little bit better. Okay, yeah. Yeah, what you see is that the light basically from the bottom goes all the way up, yeah, and then bounces around and goes down again to illuminate this light from the top. Yeah, so what I'm going to do now is I'm going to show you a little bit some of the things that you're able to do with that microscope. So over here, that's again the view, okay, yeah, and I'm going to show you now something. Look at this. Bing! You can actually have a little software here, which is a camera control. And how do you access that? Well, what you do is the following. You basically turn on the microscope, and then on the wireless, even on the mobile phone, by the way, you will see that there is a new wireless router. So this basically creates like a little access point. And then you simply type in the internet address, which is a number. It's all in the documentation, so you don't type in the basically the IP address, which is always the same. And then you can actually also access this little control panel over here. There is something, and what I can do now is the following. I'm just going to demonstrate this to you now. Look at the top one at the light now. You can see that I can I'm using now the mouse pointer. You cannot see the mouse pointer right now, but you can essentially adjust the light and brightness and contrast. Yeah, I can also adjust that. So saturation, I can go up a little bit with saturation as well. I've not tried a lot of special effects. I can't do now for whatever reason. Okay. Auto white balance enable. So there are basically possibilities to control this as well, and I can also change here. Okay, the streaming software doesn't allow me to do this now, but I can also change the resolution around and so on. So this is quite nice that you have all of those camera controls also available. So let me hide this again here, and now what I've seen is the following. Okay, now it this happens occasionally. Here we go again. Because occasionally there is a little bit of a delay occasionally there's a little bit of a delay in the wireless transmission. Okay. Yeah. These are just some algae. Okay, you see also some fibers here, some microplastics. Okay, so this is basically one thing. I'm going to just show you something else here. This is also a permanently mounted slide. Yeah, and one of the things that you will notice, of course, is that the camera is on the bottom. This basically means that let me show you here on top, the camera basically has to take a picture through the thick slide, obviously, right? So not through the cover glass, but through the thick slide. But I think, yeah, this might reduce the image resolution a little bit. But yeah, even if you turn it around, you do not see much of a difference. Yeah, so of course, this is now a little bit out of focus because the distance is different. Okay, and so I have to, yeah, but I'll just place it in like this. Huh? Yeah, so yeah, and this is a little bit the thing here. Of course, the image quality cannot be compare if you're used to the very crisp images of a microscope yeah, with proper microscope objectives. I mean, it's clear because this is not a microscope objective. This is just the regular camera lens that you've got here. But yeah, but I kind of like this system quite a bit. That this because it's so simple that essentially it kind of works as well. What I'm going to do now is the following. And now we have to be a little bit patient. I'm going to disconnect this camera now, and I'm going to correct the camera module directly. And I have to then this takes a couple of seconds because I have to reconnect wireless again. Okay. So what I'm going to do now is I'm going to try this. I'm going to disconnect this here. Okay, and then I'm going to connect this one over here now. I'm going to, yeah, and then I have to connect the wireless. I have to connect my computer. It's got to boot first, so there is yeah, a red light here. So it takes a minute or so to boot. Okay, and what I have to do then as well is I have to reconnect my wireless. Yeah, and I have to go to connect. And then I hope that this kind of works. Yeah. So let me see if this it takes a minute or so. I'm just seeing that there is a comment here, the most recent comment. I've been thinking, basically, we all have smartphones with cameras, right? So wouldn't it make sense to have something similar where you insert your camera phone with a flash as a light source? As a matter of fact, there are projects like this where you can connect a lens in front of your microscope, your mobile phone camera. So there are certain yeah, projects also like this. Yeah. The thing is, you can have two possibilities. You can either connect a small lens to the mobile phone, or you can also try and to somehow connect maybe a microscope objective. But then you have to be careful with the correct distances, and these things are significantly larger as well. So I don't know. I don't know if this is now gonna work. Ha! Look, here I am. Okay. Yeah. And what I would like to do now is, yeah, I would like to now show you, I'm going to point this directly now through my yeah, microscope eyepiece. And you see that's also one of these things I always have to, oops, now I lost it again. Maybe there, every now and then, for whatever reason, the wireless connection isn't, or maybe because I touched the connectors. This could also be a problem. Maybe I short-circuited it somehow because you should not touch the connectors. And now, see what I'm going to do now. I'm going to go refresh browser. Okay, funny. I kind of lost it. Okay. It just worked before, unfortunately not anymore. Start stream, stop stream. Or did I lose the internet connection? No, internet. Well, actually, I still have the connectors. The connector. Okay, that's a one of those demonstration effects. Okay, where for whatever reason, see, this works, and I think I might have touched some of the things here on the back, and maybe this caused it a little bit. Yeah. So what I'm gonna do is there's a little button over here called reset. So I'm going to push the reset button. Okay. Would it be easy to use as a field microscope? Yeah. As a matter of fact, the way that I see this whole project is as a little bit as a proof of concept. so that even if we get it to work somehow like this, and if we're able to connect over wireless a mobile phone, then actually we can actually, if we improve on this a little bit, maybe actually indeed have a very portable and easy field microscope. Okay, this is always the same thing. Yeah, for whatever reason. Wow, okay, I kind of lost it now again. Yeah, but I think you get the idea. But you know what, I'll reconnect this one again and see if this works. It didn't happen very often, but maybe this was one of the reasons that sometimes when you touch the connectors, I'm so farsighted. Okay, so here we go again. Where is this? I have to go here again. I have to connect again, connect. Okay, and I hope that I'm able to get this to work now. Where is this here? Yeah. So what I'm going to do is while it starts up, I'm going to read a few of the comments here. Okay. Yeah, there are greetings from all over the world, from Aargau, Lebanon. Okay, Switzerland, Slovakia. Hello around the world. Okay. But I'm trying to skip forward a little bit to the sound is good. I'm trying to skip forward to some of the questions or comments that some of you might have. Okay. Okay. Somebody likes my improvement to the setup to the live stream. Okay. So yeah, iNaturalist is very good. I use it too. Okay. Okay. Costa Rica. So you're telling us we could set up a 24/7 live stream of tardigrades at home. As a matter of fact, this is, I think, what some people are doing. You can use those small cameras and essentially, use them for permanent live streaming. I mean, people are using this for home surveillance, and then they can check using their mobile phone somehow, whether everything is okay or not. So, but let's have a look now. Yeah, see, okay, maybe we're lucky now that this works again. Okay, here we go again. It's a little bit too bright. So I'm going to turn down the, so we're up and running again. Okay. Let's have a look at some of the things here. This is a water sample, okay, that I just prepared. There were a couple of, actually a whole bunch of ciliates in there. Oh, there was a rotifer maybe, And you can see that it is not a very fast thing because there is a little bit of movement. It's not a very fast thing because the wireless connection is not very fast. But for still images, it's perfectly fine. So if you want to take a picture every couple of seconds or so, what I've not found out yet is if there is somehow a way to directly connect this camera over USB. After all, it has a USB connector to the computer, because this would probably increase the frame rate significantly. So this is something that I'm trying to still figure out. Okay. There are a couple of critters floating around here. Okay. Yeah, but you get the idea. Let me have a look at this sample over here that I have in the microscope, and that's also a fresh sample of some algae. Of course, I also have to tell you that this is the resolution is also quite low. The reason why I've chosen deliberately a low resolution and not the maximum resolution is because this way also the frame rate is a little bit faster, and for live streaming it's also more convenient. A whole bunch of single-celled algae here as well. So this is kind of how this works. Okay, I'm gonna have to watch this from the start later. This is cool. As I mentioned already, if you want to have a full documentation of this camera, there are plenty of things online that actually show how this can be configured and programmed. And if you even want to do something like that, like programming, it's not necessary, but it's possible. And I also included a link. So it's a standard, a popular standard camera module that can be easily obtained pretty much everywhere. Okay. It's sad to see microplastics in water samples. Honestly, I find them quite a lot. So if I put in a water sample from somewhere, sooner or later, I'll always find some plastic textile fibers. Yeah. Okay. I've been thinking basically we all have smartphones. Yeah, I already answered this. That would result in a much better quality connecting a smartphone, and we wouldn't have to do more expensive. We won't need a camera module, only a very strong magnifying glass. Yep. So basically, there are actually things that you can buy that kind of these are not meant to be microscopes, but macro attachments that you can connect over your mobile phone, and then you can actually go in much further closer to get macro images. Yeah, the demonstrator effect again. Yes. Yeah, the field microscope option is indeed something, and what I've heard is that some people in education and so on are already using this system. A simple smartphone camera adapter works wonders for photographing through a regular microscope. It's a bit fiddly with the adjustment, but it's really effective. I have a $12 one, which I'm happy with. Yes, so smartphone adapters are definitely the first thing that I would try out. I think it's more directed towards people who don't have a microscope at all. I would say it's more directed also towards people who want to do a little bit of tinkering around. For hobbyists and so on, and maybe also to get some maybe children or so, or youngsters who are interested in tinkering, interested in actually building something which does not cost a lot. It's a project. I really love your last video about the microbes in your coffee. I just wanted to say that I really liked that video. Thank you. Okay. Yeah, I made a video just recently where I put I found a biofilm in the water tank of my coffee machine, and it's I just thought that I made a video of this, and this was quite fun. Yeah, the module allows Bluetooth per its spec. USB 3 connection will help improve the signal discontinuity issue. The module does not seem to have the USB option. Okay. Yes, I know it does have Bluetooth. I don't know how to set it up yet. So I tried Bluetooth, but it didn't over my mobile phone, but it did not recognize it. So maybe there is somewhere after maybe setting it over the program. Maybe it has to be activated. Okay. Got here late. What are you doing today? Well, today I'm talking. I've just showed you this little microscope here, and I would like to move on now to iNaturalist. Then I'm going to answer again more questions here. Checking my mobile phone to see whether my tardigrade rates are okay would be much more important. Yep. I mean, of course, there is a limit concerning the transmission. It only goes for wireless. Wi-Fi does not go very far. Yeah. So this is simply something that you have to be aware of. Yeah. Is showing us a new and cheap microscope that we can use at home. Yeah, so basically that's a little bit simply something that I wanted to show you, okay, today. So let's move on a little bit, okay? So I'm going to switch topic if you're interested, and if you have more questions about this, if you want me to show you something, you can always post something, I will go back. And so those live streams are never really perfectly structured, so it's always a little bit improvised. Okay, yeah. And here we again have let me just put this in here for the fun of it. Yeah, that is the regular microscope that I have. Okay, yep. Yeah, so here, just some algae. So there is, I've received for whatever reason, interestingly, over the last couple of days, a couple of emails and comments from people who needed some help with the identification of microorganisms. And for this reason, I'm working on a video right now where I just show you, in a couple of minutes, simply the different types of microorganisms. However, some people also want to get a little bit also more involved in the community and maybe sharing pictures and so on. And of course, there's Reddit, which is quite popular. But I also wanted to show you an app which also exists as a regular web version. It's called iNaturalist. Okay, and it's again the demonstrator effect. Here we go. Okay, yes, you can make a free account. Okay, and essentially this is the web version, but normally you would probably use it over your mobile phone, because the point is the following: to do a documentation of biodiversity. And over the last couple of years, also microorganisms and microscopic pictures taken through the microscopes, there are more and more in here as well. I just wanted to show it to you a little bit how this works. Okay, so this is basically some kind of a citizen science website where people all over the world can document the living things, not only microorganisms, but plants, animals, rare and less rare organisms, right? You make yourself a free account, or you just log in using your Facebook or Google account or whatever you already have. And then you can start both exploring, and you can start also uploading pictures. And the nice thing about this is that it's able to use, I think, also an artificial intelligence system to automatically recognize what you upload. I think this does not work perfectly well yet for microorganisms, because maybe the database is not very large yet. But in the past, I've uploaded some insects and some plants, and it immediately was able to identify them. So I hope that maybe in the future this is also going to work for microbes. And it works basically like this, that you can, over here in the top, you can type in a search term. In this case, I did paramecium. And then you over here, you have map, you have a grid layout with pictures that people uploaded, and of course a list. And what you can see here is basically, where different people have taken a picture of paramecium, right? So it's kind of like a biodiversity study. And if you click, for example, also here on the side, then you're able to have a closer look at this, and you can read a description. There's some comments in here. Also some YouTubers. Okay. Shane's Microscope, he's also a YouTuber. You might also want to check out his channel, right? So there are some people who have also YouTube channels, microscopists who are also active on here. And I think it's a nice website to do a little bit some armchair exploring of things, right? So let's have a look at the grid again. So you can see that concerning paramecium, there are quite a lot of specimens in here already. Okay. Even look, there are even videos here. I myself did not upload a lot yet. Gotta be honest about that. And where it's been found can also be seen here. And some comments and notes. And so you see there's a little bit like a form and chat function here as well. And as a matter of fact, some of those specimens and organisms that you have here, if you specify all of the details, some of them are even research grade. You see they have this green little label here, which means that the quality of the description is good enough to be used for research. So some people actually use this also for biodiversity studies and so on. And there are also people who actually manually identify those. If the automatic system is not able to do that, then it can also be manually identified. And what I'm going to do now is I'm just going to upload a picture myself. Okay, so what I click upload over here. Okay, and then I'm going to choose files, or I'm going to just drag and drop. So I'm going to I've got here a rotifer in here, and then I'm able to basically fill out everything. So I kind of so species name, I don't know the species, but oh, look at this. I just clicked it, and it was automatically allocated the term rotifer. Okay. I don't know, I'm just going to use today's date. Location, no location specified because I've got it in my jar here. So it is is private. Okay, it's captive and cultivated. It's a cultivated water sample, right? And then here on the corner on the top, submit one observation. I think it was quite nice to actually see that it was automatically able to identify this. Submit one observation. Right? You are submitting observations without dates or locations because it did not enter the location. Observations without this information are often impossible to identify and will not be visible by default in observation searches. Continue. Yeah, so, yeah, saving it now. Yeah, and here it is. Yeah, and here you see basically what I've, yeah. You see over here this paramecium that I uploaded, okay, was not automatically recognized. But this one over here, that's the one that I've got here, yeah, was actually, yeah, identified. You know what I'm gonna do? I'm gonna upload another one over here. Let me see. I've got here, let's see if it's able to identify the Vorticella. Species name, let's have a look. Loading suggestions. Ah, look at this here. Vorticella. See, it automatically identified it. That's nice. Maybe I should zoom in a little bit here so that you're able to see this better. Okay. Today's date again. Location. Yeah, it's was in my jar, and I click submit again. Saving observation. Okay, here we go. Okay, yeah. So this is basically just a program that I simply recommend. Because some people, I received all the emails, what can I do to keep myself motivated? I'm kind of starting to lose interest in microscopy, and yeah, I've seen already everything. So how can I keep myself motivated? And I think, well, if you're able to, I don't know, make yourself a collection of microorganisms, and if you upload this and share them and document them, then I think it's also an interesting thing that you can do to keep yourself a little bit engaged here. Right. So what I'm going to do again is I'm going to go again into some of the, you know what, because microscope view is much nicer. Okay. So let's have a question. I have heard that copper acetate and copper sulfate could be used to immobilize ciliates. Do you know anything about this? Well, the only thing, it doesn't surprise me if you use copper. And the reason is because metals generally are sometimes killing off microorganisms. As a matter of fact, I did some years ago make a video where I've taken pictures and videos of a water fountain where people threw in their copper coins, because some people like to throw in money into the water fountain. And you could see that where there was the copper coin on the ground, around it there were no algae growing. And the reason is because the copper started to oxidize. And those ions, the copper ions, they apparently they block the functioning of certain enzymes. This is the reason why certain metals are actually disinfecting, or at least limit. I wouldn't say disinfecting, but they kind of limit the growth. So while I've not heard anything specifically about copper acetate and copper sulfate, I'm absolutely not surprised that they would block microorganisms, because for example, people used to use so-called silver nitrate to treat eye infections, because the silver is also a metal which also kind of disinfects and kills kills microorganisms. Also, hello Oliver, and hope you're doing well. I'm doing very well. Greetings from Mississippi. Love your content. Greetings all around the world again. Have any opinions or suggestions for seasonal microbe hunting? I live in northern Canada and we get winter and snow 6 months a year, and hard to get any plant samples. Or any thoughts on fall, spring, winter hunting? Well, it depends a lot on your interests. So if even in winter it is possible. I also made some videos on this because I mean it depends a little bit how frozen everything is. But what you can do is you can get yourself some decomposing leaves. You take them home, you leave them in a moist area for a couple of days. You allow the microorganisms to regenerate, and then you carefully scratch off the surface biofilm with an object and put that under the microscope. So there are plenty of microorganisms obviously still around, but during winter, but they need some time to kind of regenerate a little bit, especially when everything is frozen. And or you can still try to collect water samples, but you have to make sure that it stays at room temperature for a day or two for the microorganisms to start to regenerate. Yeah? Or a suggestion that I have is to keep yourself a small little aquariums or jars with water samples. Yeah, you can keep them for many months. Yeah, and then you always take something from there. Yeah? That's a popular citizen science app. I never knew that microbe component of it. Never only ever used it for invertebrates. Yes, and I have to tell you that I think there are not so compared to other compared to other organisms. There are not so many microorganisms in there. But it's been growing. I've seen that it's been growing over the last couple of years. Yeah, I love iNaturalist. Yeah, a video about giving an ID to microbes would also be helpful to many people. Good idea. Yeah. So what I would like to maybe talk a little bit about identification here. I don't know. Let's just use this one that you see over here, because this is a very important concept here. Some people want to say, okay, what is it, right? And I can tell you it's some kind of a filamentous algae, and that's it. Okay, yeah. So which species is it? Well, I don't know. At least because I'm not an expert in this. So you see, I can only stay very general on this. And if you want to know more, then you have to find an identification. You have to check an identification book. Or for example, the rotifer that I showed you. Okay, it's a rotifer. Okay, but which genus or which species is it? Then you have to dig in deeper. So the question about identification is always a little bit is okay, and what level of detail do you want to identify the organism, right? Sooner or later, you will always end up at a place where you cannot identify anymore because, they look too similar, or maybe you have to do some genetic analysis. And just because two organisms look similar on the outside does not always mean that they're that there is the same species. Yeah, there could be still different species. So you have to really the more specific you want to go, the more you have to dig in and the more information you need. But if you just want to stay on the surface, and if you say, okay, I know that this is a nematode worm, and I know how to differentiate a nematode worm from, I don't know, a flatworm, for example. Okay, and if this is at the level that you're happy with, then it's quite easy. But if you now continue to ask, okay, which type of flatworm is it, or which type of nematode worm is it? Well, then it's already becoming significantly more difficult, right? So what I'm just trying to say here with the identification, it's always a question of what level of detail do you want to identify. So a little bit like I always use the example, if you want to identify a vehicle on the road, right? Then you can always say, it's a car, or it's a bicycle, or it's a motorbike. Yeah, on that level, it's easy, right? But if you now want to know which type of car is it, or which make of bicycle or motorbike is it, then you have to go always levels deeper, and then it's always a question of at what level are you satisfied, right? Do you want to distinguish the different car brands, or are you just happy with the general term car? Yeah, and there's certain characteristics that are completely meaningless. For example, if you wanted to identify different cars, then the color of the car is probably not so relevant, right? And we have the same thing with a microscope as well, that certain characteristics are simply not always relevant, as relevant as others, okay? Yeah, mycology on there too, I suppose on iNaturalist, so there are fungi, yeah? Something very interesting I found out today, people used photo scanners for negatives and positive films to scan microscope slides with bigger specimens. Goes to show how inventive people can be. Yes, that's also something that I've tried. You can indeed also buy so-called macro. You can try to also buy macro lenses and try to photograph slides this way. And another thing that I tried a long time ago, we still had overhead projectors in the school where I taught many years ago. We used transparency foils. And if you put an onion skin on there, you could actually see the cells on the wall, projected on the wall. Yeah, because it also magnified a lot, and onion cells are fairly large, right, from the onion skin. So it's this projector method is also quite possible. Yeah. Okay, summer is obviously easy to find samples. Cannot wait for mushroom season. First year I have a microscope to look at spores. What do you do if you want to look at spores from a mushroom? You take the cap of the mushroom. So basically you have the stalk and you cut off the stalk and you just take the cap. And what you do is you put the cap on a piece of paper. And then you take something like, I don't know, a knife or fork, and you knock on the cap of the mushroom. I don't know how to demonstrate this. I'm just going to use this here maybe. Of course, assuming that the mushroom is able to produce the spore. So if this basically would be the cap of this is the most improvised thing ever, right? So if this is the stalk of the mushroom, so you've got the mushroom growing here, okay? So you take off the stalk, okay? And you put the mushroom on a piece of paper and then you kind of knock against it. Without moving it, then what you're going to see is you're going to see hopefully a pattern of spores, because the different lamellae which produce the spores, yeah, and then you basically have the different spores arranged in a nice pattern. And you can then use a stereo microscope or even a regular microscope to directly observe that. But that's a way of actually making the spores nicely visible. Yeah. Let's go back here because this picture looks a little nicer. Okay. Well, I feel like Google Lens in the best for that, as there is no greater database than, yeah, it's brilliant, cool. Search in Google for Minolta 5400 microscope slide, and you'll find a very interesting article. Interesting, yeah, because the Minolta 5400, I think is a projector, not a projector, a slide scanner. Okay. Okay. Yep. That's a great link. Thanks, Oliver. Yeah, for sure. I wonder if I could join. I'm only 11 years old. The camera module is quite affordable. It costs about $20 and will be a little fun project to customize the camera for microscopy. I think $20 is actually quite expensive. Because I know that at least in AliExpress, the top part costs about $5 or $6, and I think the bottom part, which might not be needed if you're somehow able to directly connect the power supply, costs almost the same. Yeah, so I think you should be able to get everything for around $10. Yeah. Just hope that I'm allowed to use it. Okay. Never know. Okay, I'm kind of lost at camera module. I know, I just hope that I can be allowed to use it. I don't know what he or she's referring to. Do you mean if it's okay with the website here? Right, for watching. Okay, I'm just skipping over the general comments and seeing the distribution of invasive species. Okay, that is also another thing that I think how people are using iNaturalist, because you can actually monitor over time how different species, the proportion of different species changes. I talked recently to an entomologist. He is an insect specialist, and because we're also doing in school insect projects where we do insect identification and so on. And he, every time when he finds something with our school, he actually also goes into a database and documents this. He takes some of the specimens home and he does a detailed identification, a proper one, and then types everything into the database with the purpose of actually then seeing how, in this case, climate change or other environmental changes, how this impacts the distribution of certain insects. Yeah? And I think it would also be a little bit interesting to see how this actually works with microorganisms. Okay. Just feeling and being part of a community can be a big motivation. Yes, that's actually one of the reasons why I wanted to show this app to you, because and that's also one of the reasons why I'm doing those live streams, because feeling that we're kind of part of a community kind of motivates people as well. Okay. I guess I will ask them, but I still have no water samples at the time. I need to go out and get some samples then. This is something that you can do, or what you can do if you really want something for microscopy. Also in my channel, you have to go back in time a little bit. I remember a few years ago, I think it's during the start of the Corona pandemic, I did a video about 10 things that you can observe in your household. So where I went through my apartment and I found, I think, 10 different specimens or samples that you can put under the microscope. Yeah. So while water samples are kind of fun to observe because there's always movement and so on going on, there are plenty of other things that you can have a look at. Yeah. Let me see if there's still something in here. You have to refocus. Yeah. They moved all somewhere to the side, maybe where there's more oxygen. Huh. And usually they're accumulated somewhere on one corner. Let's see if, I don't know. Nope. Yeah. Or them just, here's, I don't know. Not a lot. Yeah. There were plenty of paramecia. Look at this. Could it be maybe that some of them are already squashed? It looks pretty squashed here. I think because the water sample has been drying out now, I think many of the microbes have been already been compressed a little bit. Okay. I hope you can get an account. Oh, I see. Now I understand what they're talking about. This is because there might be an age limit to sign up for iNaturalist. Okay. Okay. It can get to minus 45 degrees centigrade, very frozen. Ooh, that's still quite cold. Yeah. I have to go. I love the stream already. I'm surely watching it. When after a couple of hours, it will automatically be available again. Okay. At the moment, I'm quite interested in more do-it-yourself staining methods for animal tissue as well as bacteria. Wondering if you've ever stained with pennings. Yes, and I made some videos about that already. Okay, red ink often contains eosin, and what I would suggest is the following, and that's something that I'm going to try out. It's Easter, and it is possible to buy Easter egg colors, and some Easter egg colors come not in a liquid but come in powdered form, and the red one contains carmine, and carmine is a stain which is used to stain chromosomes. So yeah, I'm going to go out and quickly, hopefully that there's still some Easter egg colors available, but the red one is quite interesting because it contains carmine. So they sell them at least in little plastic or paper bags with some powder, and I checked the ingredients, and there was actually carmine in the red one for Easter egg colors, and that's an easy and cheap source of actually getting the stain, because normally you'd have to get them maybe from a chemical supplies company, and then expensive, right? So PCR and gel electrophoresis machine, I heard, is good for genetic identification. Yeah, I did a lot of genetic identification of bacteria when I was still at university. But honestly, this is a huge thing. I mean, what I did is some sequencing. So basically I did not do this myself, but we had a service company inside our lab. So essentially, when I discovered a new microorganism, I sent them basically the DNA of that microbe. I simply had to extract the DNA, which didn't take so long. I gave him the little tube with the DNA, and then a day or 2 days later, I got an email with a DNA sequence. And then I could actually copy paste it into the database, and I could identify what it is. Yeah, but this is too expensive and too difficult to do just as a hobby. Yeah, it would be kind of cool, nevertheless. But that would be you need a properly furnished lab for that. There's a few ways to look for spores. One, spore prints and gill sections. That way you can show how they attach to the gill. Yep. I was going to cut off the cap off and make a spore print directly onto the slide and try that. That's also possible. Hello, I just got my first microscope this weekend, having a lot of fun looking at pond water. Yes. Yeah. I have made some great spore prints on foil paper before, so I hope the same effect will work directly on the slide. Interesting point about the genetic analysis. What sort of markers would you be selecting for? Okay, so basically it works like this. At least that's the way that we've been doing. If you want, I can talk a little bit about genetic identification. It works like this. I tell you how we've done it with bacteria, and basically it would be the same probably also for other microbes. You take some liquid. I don't have those little vials here with a bacterial suspension. So you basically isolate you've got an agar plate, and you scratch off the bacteria. So it's got to be a pure culture, only one type of bacterium. You suspend it in some water, and in this little tube, and you throw it into liquid nitrogen. It's pretty spectacular. Yeah. It's very cold, and this causes it to shock freeze, and because of the expanding water in the cell, it breaks open the cells and the DNA is released. And you keep on repeating this a few times. You thaw it up again and you throw it again into liquid nitrogen, and the water freezes inside the cell and it expands and it breaks open the cells and the DNA is released. And then what we've done is we added some, I remember, some alcohol to precipitate the DNA. And then we've added some concentrated DNA. I think it didn't take so long to do that. And then I gave this little tube, I gave that to the company, to the in-house company. And what they've been doing is they've been sequencing a certain section of the DNA using PCR. They first amplified it. It's getting technical now, but they analyzed a certain section of the DNA, the so-called the 16S rDNA, the 16S ribosomal DNA. So that's a sequence of DNA that they've agreed on, scientists have agreed on, which is very suitable for identifying bacteria. Why this section? And the reason is when that's essentially the question, what sort of markers would you be selecting for? Is you want to choose a DNA section which is between different bacteria, similar enough that you can sequence it, that you can make a comparison, and different enough so that you actually see a difference. I want to give you an example. If you choose a region of the DNA which is too similar between bacteria, and the bacteria are very different, but the region of the DNA is the same, then you cannot distinguish them. Right, and so you have to get a region of the DNA which is right in the middle somewhere, which is not too similar and not too different. And then basically that's what they're sequencing. So it's around 500 bases of DNA, and then they send you an email back, and really you have the base sequence in there, and then basically you copy paste this into the database. Yeah, but to answer your question is the region that we used was the 16SR DNA, the ribosomal DNA. So that's the DNA that codes for a section of the ribosomal RNA. Yeah. So it was a very technical excursion right now. Amazon prices the module is $20 US, which is way overpriced than your quote. Thanks for the info. So what you can do is if you want to get some cheap microscopy equipment, you can do the following. Go into banggood.com. It's a Chinese online shop. Banggood, BANGGU good, banggood.com, or aliexpress.com. Essentially it takes quite a bit of time for this stuff to arrive, but honestly, yeah, everything arrived until now. So I had actually no delivery problems from China to Europe. You just have to be a little bit patient. And you can get the stuff sometimes significantly cheaper there, but don't expect always. I've bought some adapters, some DSLR adapters, which were absolutely not working at all. Okay, so you gotta be a little bit aware that not everything that you buy might actually work. Okay, but those modules, those little modules are standard modules. There shouldn't be any problem with those. They're pretty cheap. Yeah. So if you need replacement microscope objectives and things like this, then you can. First always check Amazon, and then I see if I can find it also over Banggood or in AliExpress, and then of course returns is not realistically possible. Okay, so if you're not satisfied, forget about sending it back. Yeah, it's probably going to cost you more than getting a new one. Okay. The cell looks really squashed. This cell looks really squashed. And you know what I'm going to try now? Just for the fun of it, I'm going to add a little bit more water. I would like to see if I'm able to expand the cell again. Okay, so I'm going to show you what I'm doing here. I'm going to add a little bit of water. Okay, let's try to expand the cell again. And if it's even a cell, I don't know, but it looks like one. It's also a fun experiment. How much can you squash it before you're able to revive it again? So here's some water. Here we go. And I'm going to add some water on the side of the slide. You see over here? Yeah, you look at the top. Yeah, on the side here. And is the water being sucked in? The slide, the microscope slide, is already so much connected to the Let's try the other side. No. Ah, here we go. Let's see if it expands again. Let's see what it is. I wonder. As more and more water is drawn in, the cover glass should rise. And let's have a look if we're able to regenerate the shape, but I kind of have my doubts. Maybe some of you may have already watched one of my previous videos where I squashed an amoeba, which they're really flexible, but yeah, sooner or later even that pressure might be a little too high. Yep, well, didn't do much. Yeah, I'll wait a little bit. Okay, in the Microbehunter forum, there is someone showing a project called Microscopy of high altitude wilderness snow, which may help with getting some ideas about collecting samples from microscopy. That's interesting. Didn't read that yet. I'm going to check that out. This might actually, that's another interesting thing here as a citizen science project. If you're in a very snowy area, why not try to find? Sorry, I know it's not a nice topic. Microplastics in snow, because they have found a lot of microplastics and synthetic textiles fibers in rain. So we can expect them also in snow. And what you have to do is you have to collect the snow, and there are indeed some protocols on how you can concentrate those microplastics, and you can actually make them float to the surface by adding salt to the snow water. And you can try to concentrate it like this. I've not tried this yet myself, and I think the challenge would be a little bit how can you concentrate those microplastics, and maybe you're also able to identify other things in the snow, like certain dust particles, maybe pollen, and so on. But I think the challenge is again on concentrating it. Yeah, so the cell does not seem to become much thicker. Yeah, so that's kind of interesting. There's still a little bit of water there, so I'm going to add a little bit more. Let's see if this. Here the flow starts again. See how the other cells are all carried away. So let's see if this. So let's move on again a little bit. Interesting. Never knew about carmine staining DNA. I feel like I've tried blue inks, food coloring, which stains DNA too. Ideally, some positively charged compound. The carmine, there is a stain to make the cells of dividing cells visible, the chromosomes, and that's carmine acetate. It's called. It's carmine with acetic acid, and now the cell starts to become a little bit rounder again. There's now even some movement inside the cell happening. Look, what is it doing? Could this be an amoeba maybe? Interesting. Yeah, so carmine is indeed a common stain used for DNA staining. I have the following questions regarding DIC. Do you use Curly illumination to calibrate your scope often? Some technical aspects here. DIC refers to differential interference contrast, and that's what you're seeing right now. I just want to show you the effect, and then I'm going to talk a little about the Curly illumination. Look, there is a prism that I can, there's a little thing that I can turn up here. Yeah, I can turn this. Right? And by turning this, you're shifting a little prism. This causes the color change. So usually what I do is always have a I like blue because it kind of imitates water. And Köhler illumination is the following. Let me see if I'm able to show this to you. You see this here? This is not the this is the Köhler diaphragm. And what you do is, what I can do for calibration is I have to make sure that there are 2 screws there that I can turn so that I can move it into the center. Okay. And then once it's set up like this, I open it just enough so that I do not see the edges of the diaphragm. And the reason why you do want to do that, or why you have works with Köhler, is so that you reduce any excessive light on the slide, which might introduce stray light. And yeah, so it increases the contrast a little bit. Yeah? So the question is do you calibrate your scope often? No, I don't calibrate it often. I just look through it, and if I see if it's off center, I quickly adjust it. So it takes a couple of seconds. Yeah? Do you insert your slider prism all the way or partially into the slot as part of the adjustment technique for background color? Okay. The slider that I have here that I was turning is inserted all the time. And this is actually one of the reasons why I like this microscope so much, is because this slider at the top, which I was just turning, works for all magnifications. Yeah, there are different brands of microscopes where you have to take the slider out. Yeah, I can show you. Yeah, you can actually pull it out, yeah, and you have to replace it with a different one for a different objective, and I didn't want to do that. Yeah, so I'm always leaving it in there, and basically this allows me to then, yeah, do simple, easy microscopy without me having to exchange the slider. And the only thing is that I'm turning it, and then I can adjust the colors as I want. Yeah. And as I said usually that I like kind of the bluish color. See, the cell has now regained a little bit of its shape, but I don't know. That's what it is. So let's go up again a little bit. This is now 60 times. I have to open now the diaphragm as well. I don't know. Could be some kind of an amoeba. Maybe it's a little bit stretched and stressed. Yeah, but it's most likely an amoeba because look, it's able to change its shape. But maybe it's still a little bit inhibited in its movement. Huh? RFLP's restriction length, what was this called? Length fragment polymorphisms and SNP are the goal, but just learning it. Yeah, I'm not quite sure if RFLPs and SNPs, these markers, if they're used actually for not for sequencing, but restriction fragment length polymorphism, it's called. And I think this is a different way of actually going about it. So what they do is you're using endonucleases to cut up the DNA, and depending which where it cuts, you get a different pattern on the electrophoresis. Now I'm going technical again. But this is not what I've been doing. I've been doing the sequencing of the DNA. Yeah. AliExpress doesn't deliver stuff to Austria anymore. Really? At least most stores don't, due to the new laws that have been in effect since January, February. Oh, really? Is it the new law about the free shipping? Oh, that's interesting. Okay, I know they've started to introduce some restrictions because shipping costs from China, sometimes they were shipping free of charge, and I know that they've also now increased some import taxes. So there was an import tax limit, but I didn't know about this, that it's not possible anymore. Nice explanation. I thought the 16S was quite highly conserved, but I guess in the midpoint being not too specific and not too general. Yes, that's the reason. And if you want to sequence eukaryotic DNA, you use the 18S. 16S for prokaryotes, bacteria, and 18S for eukaryotes. Whole lot of study has definitely been done on that 16S. Yeah, that's essentially the one that I've been using a lot. Is it risky to buy direct from China? There is no guarantee that anything will work properly. Quality is out of the window. Yeah, to a certain extent, I agree with that. I mean, as I mentioned before, I bought this one DSLR adapter, and then yeah, it didn't work at all. I made a video about this, by the way. About a DSLR adapter, which is totally unfunctional. But it looks nice. It was really the quality was quite good. Look, I think this is an amoeba. Look, this was an amoeba. Now it's starting to move a little bit. Yeah. And for those of you who are a little bit new into biology and microscopy, the amoeba that you see here, that is a eukaryote, right? It has a cell organelles, it has a nucleus, and the dots that you see swirling around, these are prokaryotes. Yeah, so these are also cells, but they're significantly smaller. Yeah, so these are basically the two cell types that you have: the eukaryotes, eukaryotic cells with a nucleus and all of the cell organelles, and the much smaller prokaryotes, the bacteria. Yeah. True, but most, if not all, of the stuff you get in everywhere else is made in China anyway. Yeah, that is correct. That lots of things are made in China. However, if they are imported by a company and they're branded, then at least the company has a little bit of a quality control over the whole thing. Yeah, but sometimes things are sold as no name, and then you don't know what you're getting. Yeah, I think it's dissolving or washed away. I think it's still living. Yeah? The paper centrifuge. Okay, I've been experimenting with this, and the thing is the following. There is okay, this was my sweetie printer. The paper centrifuge. There is this thing. I've been experimenting with this a little bit, and I think, and what I've made also in the previous video is what you do is I think it works better if you want to make a homemade centrifuge if you attach something to an electrical drill. It is actually more reliable. A paper centrifuge, there are some people who've been experimenting with this. I also tried that, but I could not get the speed together that I needed to actually do a proper centrifuging. But a paper centrifuge is you have two discs, you have some discs of cardboard paper and some string, and it spins very quickly. Okay, but I found easier ways to actually make a homemade centrifuge, like for example connecting something to an electrical drill. Makes sense. Always something to do with the SNPs, then you get an ML clusters and the whole phylogenetic mess comes to play. Yep, okay. Yeah, I've been doing for my diploma thesis, I've been doing a lot of this stuff. Yeah. The middleman between helps ensure the quality with a higher price tag. For example, buying scopes from Big Four, and most of their parts are made in China. Yes, that's well, yes, the Big Four are Nikon, Zeiss, Leica, and Nikon, and Olympus. When you buy a microscope from them, and you get it configured, then they will actually buy the individual part, will assemble the individual parts, but they are sometimes imported from China, from Japan, wherever they are made. However, it is like this, that those parts are proprietary. This means for the cheaper microscopes, you have a company in China that manufactures microscopes for all different brands. And this kind of explains why sometimes the microscopes or some of their parts between different brands looks very similar. So the reason is because sometimes they are actually using the same parts. I mean, if you just compare very notable the microscope objectives, if you look at the different brands, you're going to see that you keep the same type objectives keep on reappearing over and over again. And that is basically because there seems to be a company in China that manufactures them for all different brands. However, for the Big Four, for the really expensive companies, they have their own manufacturing. So they do not fall back on those standard parts, but actually they make their own things, even if it's also made in China, but they have their own manufacturing facility there. Okay, go on, buddy, rehydrate. It says, yes, it's slowly rehydrating. Yeah. Oh, look at this. Can you see it? You should be able to see the nucleus here as well. Look, let me turn on the arrow. Yeah. See this round thing here? Could be the nucleus. Yeah. And all of those things that you see floating around here are bacteria, those tiny ones. Yeah. And those might be either some green algae or some diatoms. Yeah. Yeah. It's slowly rehydrating. Yeah. As long as it's not popped open, I think it's going to be fine after some time. Okay. Yeah, I'm going to try that. If I have success concentrating microplastics, I will let you know. Yes, please try that. What I've found online is that if you want to concentrate microplastics, that you have to add a certain amount of salt to the water to increase the density of the water, and then the microplastics will start to float to the top, and then you can kind of collect it from the top. Yeah, and that's one way of kind of separating it, because many microplastics have a similar density as the water and they won't settle down on the ground, and therefore centrifuging also won't work. So that's the reason. And what you can also try to do is you can use, and this is a little bit more advanced, you can use a dye, a stain called Nile Red. Nile Red is the name of the dye, and it sticks to the microplastics. And then you can use UV light to actually make it shine up. Might be possible to use a UV flashlight for that. Huh? Okay, great. Thanks for your response to my DIC questions. Okay, the amoeba is moving a bit. Yep, it's still not fully happy, but it's slowly regenerating. I get like 2 meters of snow at least every year and live right beside a field. Yep. Then I would say something I've not. Yeah, what you can do is the following. If it's snowing, you can try to actually put snow under the microscope, but not the actual snowflake. But what you do is you take a microscope slide and you cover it with ice cold nail polish, clear nail polish. And then you go out and you make sure that some of the snowflakes land on the nail polish. And then you allow the nail polish with the snowflake to dry. And then you will have an imprint of the snowflake in the nail polish. So you have a permanent mount of the nice crystals. If you check my other channel for that, I also made videos a couple of years ago. So snow under the microscope is quite nice as well. Thoughts on focus stacking to get some really nice combined depth of field shots? Works well with hard sectioned plants. Yes, focus stacking. I have the following recommendation. There is a free program called Piccollay. P-I-C-L-O-A-Y, Piccollay. But it's a pretty advanced program. So it has lots of configuration. You gotta read the tutorial, but you can download the free version of Helicon Focus. Helicon Focus is much easier to use. It's really you drag in the pictures and you click and it stacks it. It's really really fast and easy to use. And the trial version includes a watermark. However, yeah, but that focus stacking, I would recommend that you try it with Helicon Focus. Okay, the amoeba is moving, so is it an amoeba? Most likely it is. Yeah, because if it's changing its shape in such a way, it's most likely an amoeba. Yeah. Did anyone else see that corkscrew movement? Yeah, if you're referring to those the corkscrew movement of some of the bacteria, these are spirilla. Yeah, they're quite common in water. Okay, spirochetes. Yep. Okay. So you know what? The water's been almost soaked up now. I'm gonna add yet more water. Okay. Let's see if I'm able to. Oh no! It's floating away. Yeah, but this is actually a sign that there is now plenty of water in there, so it should be able to move. But it's not looking healthy. It's not looking healthy. Yeah, there was maybe a little too much pressure on the cell. I actually thought it might actually start to regenerate, but you see, if you look into the cell, also the cell organelles inside are not moving anymore. Yeah, so yeah, it could have. Yeah, let's wait a little bit. Now the bacteria are going to be happy because the bacteria found some food. Okay. So well done on the amoeba resurrection. Well, I don't know if I resurrected it. I don't know. Not anymore. Can you see chromosomes? That's another interesting thing. it is difficult. You have to stain them and then you're able to see chromosomes. And as a matter of fact, yeah, because you're asking, I'm going to show you. And I'm going to show you some chromosomes using a permanent slide. So what I'm going to do is I'm going to simply, if you ask, I'm going to show you. Even if it is off topic, but why should it be off topic? It's not off topic. Yeah, if you're asking something, I'm going to show you. Yes, it is possible to see chromosomes. See, I have to open the Köhler diaphragm to go down with the intensity. Yeah, of course. So these are now onion cells. Where's this? I have to find it. And it's way too bright. Okay, here are some onion cells. Where were you? Okay, here we go. It's totally out of focus. Okay, and these are stained. It doesn't say how they were stained. This is a commercially prepared onion slide so that you are able to see chromosomes. That's the whole point of this. Okay, and go 40 times. And this is usually the time when you have to be very patient and when you have to start kind of, looking for, what I'm going to do. I'm going to go away with the DIC, as I'm going to turn this off and just the regular bright field because it's, And this is also one of the activities that I'm doing with my students here, right? Yeah. And what you see over here, I can already see chromosomes. I need my arrow now. Okay, where's the arrow? Here it is. Can I not? Why can I not? Here we go. Here's the arrow. Okay, so what you see, these are onion cells, right? And each box here is a cell, but if you see those tiny lines in here, these are chromosomes. Or for example, this one over here this cell is dividing in anaphase. Those tiny lines that you see over here, these are chromosomes. Yeah? Or also over here, chromosomes, those tiny lines in here, those tiny lines up here. Yeah? So you see, yes, it is possible to see chromosomes, this is regular bright field. So if with your microscope, you should also be able to see those. But this is a commercially prepared slide. Yeah. So this basically means that there is it's been specifically made for the purpose of actually looking at the chromosomes. Yeah. Spirochetes are spiral-shaped bacteria, like Borrelia are, yes. I would be generally a little bit careful with spiral-shaped bacteria, okay? Because, some of them can cause diseases, right? Okay. The amoeba is moving notably. Now we'll have to check again, okay? Yeah, so be careful with spiral-shaped bacteria, yeah? Of course, it depends which one it is. I've been trying to make dark field filter for my microscope and them does not work no matter the size of the center I make. Any tip I get is right. I don't have them dark field filters here now. So first of all, if you want to make your own dark, here's one. Okay, I'm just going to use this here as an example. It's a 3D printed one. Okay, but you don't have to 3D print it, so you can just use your, actually, I used a blue one here because I wanted to get a blue background and not a black background. Something that is really critical is the diameter here. Okay, the diameter of the central disc is absolutely critical, number one. If it is too low, generally, the higher the magnification, the larger the disc has to be. And that's, of course, a problem because at a higher magnification, you already have a low amount of light, and then you're taking yet more light away, right? So generally, try to get it to work with it 10 times. And if the background is not completely dark, then you have to make this disc larger. If it is too large, so if you're not sure, just make it a little bit larger. It's better to err on the larger size, okay? If you're not sure, make it a little bit larger, then the background is going to be a little bit darker, but there still should be enough light from the side. And you have to completely open the condenser, and you have to raise the condenser all the way to the top. Okay, and then it should work. Honestly, a microscope that has a condenser and a filter holder, I have to yet see a microscope which does not work. Okay, but you do need to have a microscope with a condenser. Okay, does Picolet work with Mac? I don't know about that. Okay, picolet.de is the website. Otherwise, for the Mac, maybe there's some kind of Windows emulators or something like that. Yeah, but Picolet is a very advanced program, so you can even make out of a stack, you can even make three-dimensional images and so on. So it's a very advanced program. But if you read the tutorial, just by using the standard settings, you can already get a very good result. Okay. Why is the water pushing me for the amoeba? Okay, yeah. The midst of making some dark field filters, and this knowledge was very relevant. Please do check out my videos on making filters. I actually explained this a little bit in the trial and errors that I used. Now, I did a lot of 3D printing because it's easy for me to do that, easier than actually, doing this by hand. But what you can do is the following. If you do it first with a dark cardboard, and then later on, what I highly advise is because sometimes the black background is a little bit too high in contrast, you cut out some overhead foil or some plastic foil or some kind of plastic, I don't know, thing. Here is one, some plastic packaging material. That's why I kept those. Right, here, some plastic packaging material, and then you use some nail polish for the fingernails, some blue or whatever. I always use blue because of the nice background color, and then you can also try to paint something. Or if you have a printer, a color printer, you can try to print something. However, I found out that the color printers usually do not give a dark enough, okay, result. So this has to be fairly dark, okay. Okay, can you check your onion sample with DIC? Okay, to see if more details, most likely not more details visible. Okay, because, so this is now the 20 times, so let's go up with 40 times and 60 times. I have to change the thing here as well, go up with the color, open the condenser a little bit, and that is now DIC, and now when I turn this, then you see this is how there is not a huge difference. Yeah. There is not a huge difference. Yeah. So but there are here, for example, this one is these chromosomes here that you see here, those little lines. This cell is in telophase. So it's just divided, and the chromosomes will start to form loose DNA again. Yeah. That's, by the way, as I mentioned with my students, what I do is they have to look at this, they have to count the cells, and have to determine how many % of the cells are actually dividing. Yeah. This one over here is also, you see some chromosomes here and here. Yeah. Here, this one is in prophase. Those little lines are chromosomes. But honestly, just a second. Where is the Fleeta? No, there's. I don't have it here now. It's in the other, or number 22. Let me check if this is the one. Is this the one? No, that's the wrong one. I think that's the one. Okay, I think you might. Is this? Let me see if this is the better one. This is the cross section of a lily. The problem is I only can see the whole thing on the computer. Yeah, and not. Yeah, here we see some nicer chromosomes, if we can find them. That's my standard specimen sample. Let's go up here. Some of you might have already seen those, because it's this one that I've already made a video on. So, where is it? Yeah, here it is. Oh, I bumped into it. I bumped into my stage. Here, let's go up 60 times. Yeah. So that's basically also plant a cross section of a lily. And here you see these are chromosomes, those lines that you see here. Okay. And I'm using, DIC right now. That's why the colors change. But you see there is not a huge difference. Yeah, so you see that not every technique is ideal for everything. Could I say anything on Lycopodium spores? Sorry, I cannot say anything about those because I don't know them. Okay. I assume that you already own a microscope objective that is made for dark field. Actually, you do not need specific microscope objectives for dark field. The regular objectives work also for dark field. The only thing that you need for dark field is a filter like this, and of course a condenser where you can put it in. Okay, dark field does not need specific objectives. That's you just use your regular objectives. Dark field is when the light strikes the specimen from the side and then is reflected into the objective. You do not need dark field objectives. They don't exist. Okay. I used Tinkercad and designed some patches of various sizes. Okay, and then used my college's 3D printer to print them out. Yes, as a matter of fact, that's how I've tried it myself, because it was simply much faster to actually make those using a 3D printer. Okay, thanks for the video. Oh my gosh, it's already 1.5 hours. Why I made my own dark field filters. It works for the 4x and 10x objective, but the background is white for the 40x and 100x objective. Yes, because the central patch stop was too small. If you want to get a dark background for the 40x, you have to make the patch stop larger. Okay. Large enough that it will basically make the background dark, but because this reduces light overall, it's gonna be generally darker. Okay, but I've actually managed to also get a very nice dark field also for the 40x. I cannot believe it is you. I did not see you for the whole live stream. Okay, maybe a little. Okay, maybe somebody else. Nice pink stain for the lily. What is the easiest way to connect a microscope to a monitor or computer? Depends. What you can do is the following. Let's do it like this. There is you can use a mobile phone, and you can connect the mobile. You can install a software on the computer which uses your mobile phone as a webcam. Okay, so this actually works. There are a couple of programs available. And then what you can do is you just connect your mobile phone. If you want to use that, you connect your mobile phone using a mobile phone adapter, and then you have a USB cable or possibly even wireless. And this USB cable actually goes into I prefer USB because it's more reliable. And then you have essentially an app, a program on the computer, and the mobile phone is like a webcam. That's basically number one that I would suggest. Second possibility is you try to connect the webcam directly, or if you have a DSLR like I have, like a Canon or Nikon DSLR, then they have an HDMI out. And this HDMI out can be connected directly into a monitor without the need of a computer. So that's kind of convenient as well, to not have a computer. You just use it directly. Sometimes the image quality is not so high because it depends a little bit on the quality of the HDMI signal. Or you can also connect, many DSLR cameras have that, is you can also use the DSLR output of the camera to connect it to the computer. Okay, so this is there are different ways how you can do that, but I would first and foremost try out a mobile phone and try to download a program to install on your computer so that you can use your mobile phone as a USB camera. Okay, I would say that is one of the easier ways. Thanks for checking the sample with DSC. Scanning electron microscopy seems to be one of the good options to view the details. Yes, but this requires a lot of specimen preparation. The law regarding AliExpress is to do with packaging. The sellers have to have a person in Austria who disposes the garbage. What? Really? That's an interesting. The sellers have to have a person in Austria who disposes of garbage. Thanks for the info regarding dark field. Interesting. I suppose this has to do with import issues or so. I don't know. But for dark field, you have to fully open the diaphragms and give full illumination. It does not work at all on all magnifications. Yeah. Okay. About 90 minutes of live streams now, 93 minutes. And I think I'm slowly starting to feel tired again. So if you joined in late, I just want to give a short summary of what I talked about today. I talked today about those little do-it-yourself microscopes using those standard cameras here, which I think are interesting bases also for experimentation, especially because they're so cheap. Okay, so that was number one. Then I also introduced to you a little bit the program iNaturalist, which I recommend that you have a look at if you want to kind of share pictures, or if you want to have some do some identification, for example, or if you want to feel being part of a community. It's also kind of helps a little bit. And then I did a little bit of question and answer sessions like every time. There are a couple of final comments here. There are special condensers available for dark field. There are benefits. The special dark field condensers that some companies offer, essentially what they have is they sometimes have built-in illumination, or they have some kind of rounded mirrors which kind of make sure that the light bounces off, so you have a much higher intensity. Okay, so those dark field condensers actually are yet better than just using a patch stop, but not all companies have those. But the patch stop should actually work with pretty much any microscope. Yeah, and it might be actually good enough for most cases. Yeah. The garbage from packaging, cartons, and styrofoam. That's interesting. Okay. Cable length and type of cable for HDMI will have an impact on image quality. That is correct, but also the camera, because what I read is not all DSLR cameras will actually output if high quality HDMI signal. They will only output the same quality as you see on the little screen. Okay, so it depends a little bit. You have to look for something like clean HDMI, they call it, which is without all of the additional menu and so on. So some cameras have this concept. It's called clean HDMI out, which gives you a high resolution HDMI, which you can actually use to actually do further image processing. Yeah. Okay. Just try dark field for some plant sections I prepared a couple of days ago. Nail polish and glycerol permanent mount and first impressions are very cool. Thanks for the motivation and tips. Yeah, and what I would suggest is once you got the dark field sorted out, then do change the color around. Especially for water samples, this is really nice because the contrast is not quite as high and you get a nice blue background as well. And you know what? Because it's 4:05 in the morning in Vietnam right now, and it's 11 o'clock at night where I am. Okay, is something where I think I'm just going to leave it at that. I hope I was able to motivate you again a little bit in trying out a few things concerning microscopy. If you have any suggestions, please to post them. Also, if you joined in late or if you had to leave, then yeah, the video will be available again online after some time. And I'm going to say bye-bye, all the best, happy Easter if you're celebrating Easter, okay? And I wish you all the best, and happy microbe hunting as always, and see you hopefully next week again. Bye-bye.